Yes, so once more, welcome everyone. And without further discussion, I would give floor to Sharif Islam, who is presenting DiSSCo for us today. Please, Sharif.
Yes. Good morning. Thank you for organizing this. And as mentioned, I will be talking about DiSSCo. Can you see my slides?
Yes, it's perfect.
So DiSSCo is a distributed system of scientific collections. So today I will briefly give you some high-level overview and some technical introduction. And if you're interested in more details, here are a few links, our webpage, blog, and some of our activities in GitHub that you can follow along. And I'm currently working as a data architect at NATURALIS in Leiden. NATURALIS basically leading the DiSSCo project at the moment with several other European partners. So what is DiSSCo? The goal is to digitally unify the fragmented landscape of the natural science collections. And when we talk about natural science collections, we mean all the natural history museums, herbariums, and different other research collections around Europe. And these collections host all sorts of physical objects, specimens, collection materials, manuscripts, and we've been doing that for the last 200 years or so. So there's enormous amount of objects there, and these objects not are just for display, for education. They're amazing scientific resources for not just biological science, for all sorts of different disciplines. So the idea is sort of digitally get this fragmented landscape together under one curation process, policy, and practices. As you can see here, we're working with 170 plus national facilities, and more organizations are interested to join our consortium across 23 different countries, sort of to create this one big virtual European collection. Why do we need this? So the reason this project came about, and we're also in the S3 roadmap, which is the roadmap for some of the research infrastructures. Of course, digitization and some of this IT related things within our domain is not new. We've been doing that for a long time. However, the current model in a way is slow because all of these physical objects need to move around for various research purposes. The researchers need to move around. There's lack of coordination and fragmentation within how some of this scientific data moves and scientific data request comes. And of course, a lot of this physical access limits the way you can do things in a broader scientific disciplines. So we want to change that in an integrated research infrastructure model, provide wider access, and this integration hopefully will lower costs and will make research procedures faster, provide new insights with all this digitization and digital models, and of course, fair, findable, accessible, interoperable, and reusable is also a key element to this integration. So where are we now? We're currently in our preparatory phase. We're not a ERIC yet. We're still a Horizon project. And we're going towards our sort of construction and operation phase in the next few years. However, there are a variety of different related projects that has been happening in the last few years that's been producing different services already. We have a lot of prototypes and demos and proof of concept coming in, and of course in different European projects when we're working, we're also producing things that will go towards the big research infrastructure preparation. So a lot to look forward to in the next few years, but also a lot of things happening, but I'll talk about those in a minute. So here's the sort of the big concept that we're working with, and this is also in a way similar to the digital twin type idea. As I mentioned, all these physical objects we have in our collections around Europe, almost the estimate is 1.5 billion physical specimens. And of course, digitization is happening, but we want to take that digitization, analysis, and computation to the next phase with a fair digital specimen. And the idea is that not just have a digital entity or digital representation, make that digital representation an actionable knowledge unit. And I'll show an example what we mean by that in a minute. So it's more than just creating a web page, more than just getting a picture. We want to connect it with different data sets, different types of operations, and expand this collector of this information in a different domain. And this is happening in the conceptual planning for this happened in the last few years. However, there are policies and standards in place that helps us, curate the physical specimen data. There's already a variety of different data standards. There's different ways of digitization and workflow. All of these things, with the existing practices, we want to bring it to sort of next level in this digital object architecture level with the idea of digital specimen. And particularly, I'll give an example in a minute about how we use the persistent identifiers and metadata for all these objects to connect them with other data classes, workflow, and doing different sorts of scientific operations on them. So the key thing to remember is that, of course, we're not starting here from zero. We have histories and policies and tenders that we can reuse, but at the same time, we need to think about the next levels of operations and policies as well. And one way we're doing this is a sort of a wide array of global consultations and discussions. We started with our concept of digital specimens several years ago. And at the same time, from the research domain, especially from the US, there was a lot of talk about digital extended specimens, and particularly when you collect an object, a sample, how do you collect data, look at the object and the extension of those objects around it. And from all those discussions and also the discussion around fair digital object architecture, we came up with sort of this conceptual framework of how to think about digital specimen and digital extended specimen. So this is a very simple abstraction here, and there's a paper recently came out. You can find the link here as well. And again, the idea is to describe these physical objects in the digital realm with a persistent identifier and a specific type of metadata. And I'll give an example soon, where you'll have some core primary specimen data, but at the same time, there will be derived information, associated information related to the specimen, also media images and things like that. And the whole idea of extension is that we take these physical objects and take it to different links and different operations that might be related, derived, or not stored in the same place. So there's also a distributed aspect of that. However, this PID, this persistent identifier, is the key to get you to the information you need. And of course, there's always this connection with the physical object that we have in our collections. So this is, again, similar to, in a way, you can think about a digital twin as well. The physical and the digital sort of connecting together, or at the same time, the digital realm is also being evolved and dynamic. And we're building these digital specimens and other things, and we're using them as building blocks for the services that we're planning. So at the bottom, you see the physical specimen and the digital specimen and the details for each of them, each of the objects. And then, of course, some of these objects are aggregated in different fashions, either through geological time or preservation, different dimensions of the collection holdings. And all of this provide a structural overview of all these collections that the institutions have. And we want to do that in a wider European collection fashion. So quickly people can find out what has been digitized, who has what, in what digitization level, and how that connects to other different types of services, not just DiSSCo, other services as well. So some of the DiSSCo services we're looking into will have integrated entry point for people to find these objects and the digitization level, and they can request them. They can also help them to curate and annotate these things. There's will be a lot of different types of data refinery system, dashboards, knowledge base, and of course, different sorts of new e-services will be coming along based on these components. Here are a little bit more details about some of these services, what they will do, and we can categorize them in three sections. Of course, we have the objects and the derived data. There's also people and access, both physical and virtual access, and of course the organization and the structure that connects all of these things. So on the specimen side, as I mentioned, there will be a repository of all these specimens that will have the persistent identifier connected with that, and we're already doing a lot of design work for that system in a separate European project, but well connected to DiSSCo and several other international partners. Basically build a system where each of these digital specimens will get a DOI. And of course, after DOI, after the metadata, we want these objects to be dynamic. We want human experts and machine to come and enrich this data, annotate the data, sort of a wider community curation model. And those annotations we also want to store as fair digital objects. And that's kind of makes that specimen data much more interesting and dynamic. But you need people to do that. So there are systems we're already working on where people can come and register with their ORCID ID. And there's authentication and authorization system we're working with that will help people to use single sign-on and federated authentication. At the moment, the ELVIS system is used to register transnational access and virtual access calls and requests. So these are the services currently happening through various European grants where researchers can request to visit a museum or collections. And then the review panel looks at those collections based on the capacity and resources and grants those access. So at the moment, these are happening through grants and calls. However, within the scope of DiSSCo, when DiSSCo will be operational, this will become a full-fledged service. And the way we are looking into identifying this organization is through another persistent identifier called ROR ID, that uniquely provides a link to the institutional metadata, and of course takes you to the institutional webpage. Because oftentimes we see different ways of describing the institution that creates problems, so we want to solve that with these ROR ID as well. We're also working on documentation, knowledge base, and of course there's a lot of data modeling works happening behind the scene to make all of these services work. Here's a quick example of how data enrichment could work, in particularly in a automated and machine-enabled operation. So on the left, you have the digitized image of a herbarium specimen. This is a leaf. This is a very typical way of preserving these materials, and you have all sorts of important data and labels around it. And of course, each element within the images can have a lot of interesting information as well. So this is a very useful and interesting application of machine learning systems. So in this example, you can see that it has some sort of deep neural network operation that can detect plant organs or specific sizes and width of the leaf and other important information, and it can label them. That can be helpful, of course, for other types of investigations. Usually, a typical first level of digitization comes what looks like on the left-hand side, maybe just with the label and the collection information, not much detail. There might be text around it, maybe in a paper. However, we also want to connect them, and we also want to make them as automated as possible within the digitization pipeline. So these are the sort of services that will be coming along. Again, these could be also very interesting for the digital twin application as well. And to look at the little bit of conceptual elements behind this is one way to think about it. So all this colored box are coming from our data lifecycle and data management plan. Even though here you see in a more layered fashion, you can think of it as a sort of a circular thing. And again, the key is on the bottom, we have the physical realm, and then the digital connected through lots of variety of different workflow and data processing ontology. And our sources of data comes from the collection management system through the digitization process. At the same time, we curate, publish, process, and use all of this data. There are taxonomic data, images, sequence data that coming in, but there's different types of data modeling and ontology that we have to sort of consolidate in a standard fashion to have this thing work. So that's where the fair digital object come in the digital specimen level with the persistent identifier, specific type of metadata, predictable resolution behavior, and make them as machine actionable possible, so that on the top level you have the linking and enrichment. You can do all those exciting things with that. So this is sort of a conceptual model, but again, thinking about the data lifecycle from the beginning of the specimen. As soon as we digitize, we want to start curating, processing, and use them and create values out of these data sets. We're also looking into specific serialization of this data, JSON and JSON LD in particular. Of course, there will be options for both humans and machines to grab this data in different formats through our API. This is a simple visualization of one of the JSON transformed into JSON LD, where you can see on the right-hand side, we're already using some of the data elements from Darwin Core, which is also used by GBIF and other partners. So there are some alignment within that level, but at the same time, we also have our open digital specimen data, which again connects to the digitization pipeline and digital specimen concept. So we're combining some of these things in this representation. A little bit more detail about the digital object. So this is an example of what we call an annotation digital object, and some of the data model element we borrowed it from a W3C concept. So on the top, you see the PID, the handle for the annotation object, but this annotation object is actually connected to a physical object. So basically, the arrow pointing to the target in annotation W3C model, target is basically the thing I'm annotating about. So here, the specimen has been digitized. It has its own PID. I go in either as a human or a machine, and I do some sort of annotation. In this example, my annotation is a quality flagging. You can see here the motivation of the annotation, which is again coming from the W3C model. And then I provide some sort of details, either comments or text about what kind of quality flagging is this. This could be a comment, this could be an error connection. In this case, I used a data quality check from a community standard, and I have a link to that, and I have some sort of scoring for that. And then the W3C standard also tells how to capture who did what. So they have a separation between a creator and generator. And of course, we want to capture the time stamp as well. So this is a very simple example. We're still working on the details of that, and we'll have some demo in a conference soon about this. But the whole idea is that this connection between the annotation PID and the digital specimen PID should be connected. So I can point to this, go here also from the annotation PID, I can find out which digital specimen is related to. And in a way, this annotation could be also sort of a provenance, even though this is not the provenance data model, but you can also figure out from this who did what, when, and why. So that's also important for some of the workflow and processes. This is also an example of the detail of the digital specimen, because that's a different data model. Again, the persistent identifier. MIDS is a standard we're working with for the minimum information of the digital specimen. So there's a way to calculate the number, the version of the PID when it was created. We're also working with the different types of specimen, because as you can imagine, all these billions of specimens spans in different domains. So each of them might have different types of metadata. Of course, the local physical specimen ID, the name, this could be a scientific name for a biological specimen, but this could be also a rock or mineral. And of course here, this is the raw ID I mentioned before, uniquely identifying the organization with the identifier. And then from here, you can also find out more information about that. And there are original data that we harvest that could come from different sources. Here we have Darwin Core information, probably coming from the collection management system that we harvested this information. So again, idea is using this persistent identifier as an anchor, so we can do operations like this, either through human or machine agents. Okay, so I think this is my last slide, just to keep you updated about some of our upcoming events. There's a conference happening in Sofia. This is the Biodiversity Informatics conference called TEDWIC. We'll be presenting about our work on the persistent identifiers and the annotation system, but we will be also talking about some of our output of the Bicycle project, where we're working with eLTER and a few other research infrastructure, LifeWatch and GBIF, to connecting the dots between specimen sequences and literature, and taxonomy. We will also have presentation from 2 national initiatives from DiSSCo Flanders and DiSSCo UK. They will talk about how regionally they're working on similar things with the regional partners, but also at the same time thinking about the European landscape. And then in Leiden, also later this month, we have a conference on the fair digital objects. Again, the fair digital objects is a core element for our infrastructure. And the idea is also will be used in digital twin BioDT as well to think about data interoperability. So we'll have 2 presentations talking about the data infrastructure and connections of fair data. And we're also working with GBIF and DataSite, a potential pilot to look at, again, those PIDs that I showed earlier, but at the moment these are handle, which is similar to DOI, but different type of PID. And we want to use these systems and use this metadata to create DOIs, but with the help of GBIF and DataSite. And the target is to register millions of these digital specimens as a pilot and see what are the problems and what kind of feasibility issues we have. So that's will be coming up. You'll hear about that more on that soon. Yes, I think that's my presentation. Thank you.
Thank you very much, Sharif. That was, yeah, clear and interesting. Are there any questions? You can use the raise of hand function to ask questions. Yes, Carol.
Yes, hello. Thank you very much for that, Sharif. I was just going to also mention that eLTER are also working with DiSSCo on the Synthesis Plus project on building the computational pipelines using the eLTER computational tools infrastructure, where we'll also be building research objects of the workflows themselves, not just the specimen itself. I'm sure Sharif meant to mention that.
Yes, yeah, definitely. And that also, we will be also talking about that in the work package 5 in BioDT. So that has a lot of overlap with the digital twin project as well. So again, the idea is that all of the specimens, when we start with them, has very bare bone information. So we need lots of automated workflow processing to make them enrich the data. So that's where the eLTER specimen data refinery project will come in to automate these as much as possible. Yeah. And they have a lot of experience with workflow operational and orchestration, and we will use those expertise.
Yeah, okay. Then Ute, please.
Hi, Sharif. That was very interesting. Thank you. So you talked about metadata. Are those metadata standard parameters for all the different samples, or are they just based on sort of groups of samples or specimens? Sorry, not samples.
Yeah. I mean, sometimes we do differentiate between samples and specimens. Yeah. So there is as I mentioned, Darwin Core is sort of our big standard that both used by DiSSCo and GBIF and several other research infrastructures. So that basically is in a way simply describe the specimen objects sort of in a very high level. So it describes what it is, where it came from, when it was collected, and it has a lot of other information, but you start with that. And then for each subdomain, it gets a little bit more complicated. And that's where some of the standard interoperability work we will have to do. So for example, a botanical specimen has very specific way of describing things than, say, a fossil or paleontology specimen. It might be coming from the same museum, but there are elements that are common, but also there are some extra elements that we have to describe.
Yeah, I was more thinking, do you also describe, for instance, the soil type and the atmospheric composition, or sort of like, is it a polluted site or a clean site?
Not necessarily, no. I mean, it depends on the way it was collected. So primarily, the collection is oftentimes focused on the object itself. I mean, there might be some information about where it was collected and when, but not necessarily the sort of the environmental. But this is changing sort of this idea of extended specimen coming into change that, for example, you might collect some eDNA sample at the same time you collect your specimen. And your specimen might be curated, but your eDNA sample might not be stored forever, but you still have those data connected to the physical objects. For example, water sample or soil sample mentioned, it might come, but traditionally, you know, the museum curation focused on sort of the object that needs to be curated for a longer time.
Thank you.
Okay. And Jana, please.
Yes, thank you, Sharif. Really interesting presentation. I was paying attention to the access that you mentioned in the beginning. You said that you have now, of course, we all have these transnational access schemes that the European Commission provides. But then you said something about bringing the access to a different format or creating access tools. Can you clarify that?
Yes, so this is coming from the Synthesis Plus project that Carol mentioned earlier. So one of the outcome of this project is a system called, and I can put the link here, ELVIS. And this is basically a one-stop shop for researchers to request access, transnational access or virtual access. So you come in, you know, you log in, and then at the moment it happens through different calls. So you see what calls are open, and then basically you apply for this call. So at the moment the ELVIS system is being used for that. So we created a system where you can look at the details of the call, and then you can register your organization. You can also add another organization that's going to be participating with that. So sometime you might have to visit more than one site. So in your request, you can say, okay, I need to visit Museum A, Museum B, and I want to include that in my call as well. So what we've created is that we have a list of participating organization in this call with specific identifier and specific details of that. All the specimens and collections that they have, we have that in a separate system and scattered way at the moment, so that's not connected to this system yet. But the idea is that when users go here, they can see everything, and then they can request specific things that needed. And we are also thinking about lab access, other facilities. So they might have to work with the genomics lab while they're visiting. So you can also request those access while you're doing that. So in a way, at the moment, each museums and each organization have their own systems. Oftentimes this happens through emails. So we want to have these sort of in a integrated fashion with the researchers' ORCID ID and the organization ID connected to that. So when something gets published, we can point to these proposals.
Thank you.
Yeah.
Thank you. Johannes, you had the question related to the ROR in the chat. Do you want to ask it still, like also out loud?
Yeah, it was just because that's one part we also thinking of. We have this time system. You know, we are having persistent identifiers for the observation locations, because in the context of eLTER, this is quite important. I will come to that in a minute. One thing we always also have about are these research organizations. So that's why it was quite nice to learn about this ROR idea. I wasn't aware of that. There was already this answering. One question is on how persistent is this ROR idea? Is that a project ongoing? Is there some infrastructure behind which ensures the persistence of these identifiers? Because basically that's one of the interesting interlinks also between the different research infrastructures, or also research projects is enabling, for example, this linking and using the same identifiers. So
yeah, no, that's a very important question. So yeah, there is a consortium behind it. So I think it originally started with some of the people that are involved in the ORCID ID. So it's in a way very similar organizational model. So it's not like a DOI foundation, but they are, kind of, going towards that kind of model. So there is a group of body behind it, and you can find out more. And previously there was another organization called GRID. So they have kind of captured some of the data that's happening in that system as well. So there is active development going on, both from the technical side and the organizational side. But at the moment, it's mostly on the organization and department levels. I haven't seen anybody using it for the way you would use the DIMES ID for vessels or sites. But maybe in the DIMES you can point to the Aurora for the organization that's part of the site. So that could be interesting.
Yeah, because that really would be this interesting part, because at the moment it's also this funding organization or funding bodies for long-term observation sites. How you do it by the name and the address, but having, for example, a kind of linked data component. So the persistent identifier, this really would be quite nice, because it really enables this linking.
Yeah. So we're working a lot with ROR. So if you're interested, we can also talk more about it and show how exactly we're using it. Yeah.
Okay, thank you very much. Thanks, Sharif, once more. And then we will move from DiSSCo to eLTER. So Johannes, please.
Yeah, just give me a second. So just need to, thanks. Basically, what I want to do is to talk you through the eLTER in the next couple of minutes. eLTER is one of the research infrastructures participating in the BioDT project, mainly on the part of data provision, but not only, but also in the data model development and implementation. So the colleagues from UFZ, we have Jana here, we have Ute here. So any questions? So there is some backup with what I'm talking you through. My name is Johannes Peterseil. I'm from the Environment Agency Austria, leading the team on ecosystem research and environmental information. And in the frame of eLTER, in the eLTER project, I'm participating in the information management, work packages and activities, and mainly focusing on the requirements. And that is also why it's quite good to be here, and also to learn a bit more about the use cases and what we have to deliver in the future. eLTER, the eLTER is in the phase similar to DiSSCo. So eLTER entered the S3 roadmap in 2018 and is now in the preparatory phase with basically two projects supporting this process. There is the eLTER Plus project, which is focusing on the prototyping, piloting the services, and also gaining an insight on the scientific value, and the eLTER PPP project, preparatory phase project, which is working towards the governance model of the eLTER, which should be operational 25 plus. So what we are aiming. As Jana already mentioned, there is also this VATARA model, and I will come to that. What I'm like to talk you through is basically three parts. There is one on the background of eLTER, what eLTER is about, and what to understand a bit more. Because also the question of sites, Sharif mentioned site in our perspective, it's really an observation location, not a museum, but it's really the place where things are collected. Then a bit about the technical infrastructure we are doing, and the last part, some slides on the relation to BioDT. What we are trying to do. So what is eLTER about? So in order to understand the effects of global change and climate change on ecosystem processes, it's needed to have a quite a holistic approach in collecting information simultaneously on ecosystems. So eLTER, the Integrated European Long-Term Ecosystem Critical Zone and Socio-Ecological Research Infrastructure, which is quite a complicated name. So that is why we normally just refer it to eLTER, is a site-based approach. So it's really focusing on catchments or on ecosystem types. So it's with a defined area. It's long-term observation on research, focusing on a whole system approach, and they will come to that in a minute. And the important thing, it's covering a quite broad biogeographic range. So it's really covering Europe in its geographic terms. So starting from Israel to Sweden in the north, and from Portugal to Slovakia in the east. So it's really covering all biogeographic gradients we have in Europe. It's addressing key drivers of change. So it's focusing on this input and output variables, but also focusing on the change. Just a second, on the change of the ecosystem itself, and addressing the major European ecosystems and socio-ecological systems. So that it's not only in natural habitats, but also covering agricultural areas, even urban areas, and it's addressing also this social, this human nature interaction, focusing on the LTSCR platforms. So the main aspect, it's ecosystem function, biodiversity, and ecosystem services, and that's a quite nice link to what BioDT is going to do. eLTER, the eLTER sites have a long legacy. Most of the sites originate back to the early 90s, where the forest died back, but there are also some other sites which have a longer history, even before. So we are building on a long time series of data sets with legacy data. So that's a bit similar to what DiSSCo has, with all its problems of harmonization and also access to the data or the information. And this is also what the eLTER approach what the eLTER tries to approach. So conceptually, the background is the Wales approach, and Ute, I think, will be able to talk more about that, what stands Wales for. It's the whole system approach for in situ research on life supporting systems. So covering on all these elements of an ecosystem, and it's combining with this orange ellipse and this upright. So it's a combination of horizontal and vertical components. The horizontal component stands for the interaction between the environment and the human use of natural resources, and the vertical components indicates the interaction across scales. And for both, there are different concepts we were using, and basically eLTER selected the pressure pulse decay model by Collins for this horizontal component, and this macrosystem ecology for the vertical components. According to the integrated Wales model of eLTER, aims to investigate this human environment interaction at the scale of in situ sites, so that's on catchments or regions, through their nested design, and covering also this continental scale through representative composition in its distribution of the network. So that's the conceptual background we are doing. And when translating this to the array design, so each of the categories is following this whole system approach. So what I said, so it's covering all the different aspects. So from the geosphere to the atmosphere, including the social ecosystem, the social ecological ecosystem. So this human nature interaction, and aiming to provide a set of services to research and to different stakeholders of the research infrastructure. So that's the main element is this basic site infrastructures, which are sustainable, which have this long history of data, but also providing then services on top of it with data nodes, integrating baseline standard observations, which are really harmonized across these different sites, but also linking to existing other data sources, what we call this information cluster. So really provide this information for scientific research to perform them in future. And it's mainly about this access to sites and data through a central service portfolio. In the long, eLTER aims to build on to about 200 innovation hubs. These are not newly created sites, but it's building on a legacy of about 520 eLTER sites and LTSR platforms in Europe, which are which are there. What are the main scientific questions? So we have now this conceptual design, which is this Wales approach, and the main questions, and that is also a nice link to what BioDT does, are these great challenges the LTRI tries to address, which are also linked to major European policies. And that ranges from biodiversity dynamics and focusing on biodiversity loss and the effects or the reasons for biodiversity loss, to the effects of climate change, but also eutrophication and pollution and its effects on ecosystems, and on this human nature interaction in terms of usage of resources and sustainable management. So there the focus is really on, for example, nature-based solutions or sustainable management. In order to streamline these observations, eLTER is focusing on the definition of standard observations and standard variables. So that's one line of harmonization and integration, and that's a process which is ongoing at the moment, which allows in the future to develop what we would call eLTER standard data products, which are of added value to a greater community of users. How will eLTER be organized? eLTER will have a kind of a head office and topic centers, which are providing also this the central services, which is a service portal which enables the access to the components of the eLTER. But, and that's a very important part, it's also having this site part, so this national infrastructures with the LTSR platforms, which are regions up to 7,000 square kilometers, and eLTER sites, which are normally at catchment scale. And this central service portfolio provides the access point to the users. So the main services to provide will be access to the physical infrastructure on the one side, using research sites for experimentation project, whatever. But there is also the second part, this access to long-term in situ data, which are, going backwards using the legacy data, but also in future having a higher standardization through the standard observations. There will be services on data and analysis, on analytical tools, on quality control, so to support data workflows in the research infrastructure. And there will also be a component on the support, training, enhancement, and development of future research technology in the future. So that is what eLTER is providing, and for a number of different stakeholders, where the research community is for sure one of the important ones, providing this access to site and data. But there are also these internal stakeholders on this harmonized workflows for data curation, quality control. But there is also this bigger part, which is from environmental policy decision makers, which are using or should use the results of the eLTER in the future. What are the tools and services we are building up so we are similar to DiSSCo in this preparatory phase. We have this research project ongoing, doing this piloting and prototyping of the different services and building them up in order to enable a fair data flow in the future. There are a number of existing components we have. The important part for the eLTER is the eLTER is a distributed system, so building on the national research infrastructure, which you see on the bottom of the slide, which is the main building block for the observation in the data acquisition and data provision, which are then managed through a number of central services, which can be then accessed through this ERI service portal, which will be this single entry point for the user community of the eLTER. So there are different components which are existing. We already mentioned DiSSCo, which is the main registry of site, so which is this observation context, which enables this linking. We are using also persistent identifiers there with the DIMES ID. There is the component of documentation, the digital asset register, which is the main catalog for the eLTER data products. There is a visualization component with EcoSense and the data integration portal, which enables to visualize data. So there is a prototype available on that. And the main part, there is this data layer, the central data node, which manages data from the eLTER according to different data processing levels, which are from quality control integrated data up, and that's something we develop at the moment to synthesize the eLTER data products, which should integrate data from a wide range of sites. There is a component which is called the eLTER data labs, which allow to develop analytical pipelines and data processing chains. And there it's also a quite important point what we are doing also in the BioDT project in work package 5. It's on how these workflows also can be documented and described in a fair manner and linked to the data products, which is the part where we are working on provenance and provenance model on how to link this information. In addition, we are also using services from the EOSC context. So namely at the moment it's UDAT services for intermediate data products and data collections. So that's B2Share and B2Drop to have some places to collect information. In terms of semantics, we are developing the EMFTES vocabulary and the eLTER controlled lists, which allows this annotation using an ESCOS vocabulary in the different metadata, but also on the data level. Times SDR is one of the central components in the management of the site network. We have in the eLTER context, it's about the documentation and the description of the physical observation infrastructure. So that's the site on the one side or on one hand, or the LTSCR platforms, but it also goes down to observation locations, to sensors and so on. So that's really this documentation of the physical infrastructure, which then can be linked to the different data sets. What we do through the DIMES ID, which is a persistent identifier to allow unambiguously identification of these components in the network itself. What we are doing now at the moment is this prototyping and combining key elements of this eLTER IT components to provide value to the users. So that goes from the documentation of the sites to this visualization of information and access to data to have a kind of workflow upstream to do that. It's also about mobilization of current observations, so which are ongoing, but also going back in time in mobilization of legacy data, which in terms of eLTER are both biotic and abiotic information, which are collected on the different eLTER sites. So where do we see this link to BioDT at the moment? So I had a quick look on the different use cases, and I think the LTRI can provide information to most of them. Basically, also to the genetically detected biodiversity, which I haven't marked here, because they are especially information on the environment, on abiotic characteristics is important. And so I think this is what eLTER really can provide on the level of the different sites to have this combination of, for example, like deposition, soil characteristics, but also runoff, which is quite important for the different models, but also like on soil physics. What we did, and basically that's I think also similar to what DiSSCo or other research infrastructures did. We are checking on the capabilities of the eLTER infrastructure. We looked or we had contact to the different contributing sites and looking into what kind of data they are able to provide, on which temporal and spatial resolutions for continuous observations or sensor-based observations we have here, from air quality to soil, water, or so to the different components of these life supporting systems, and also to the what we called plot-based observations, which are with a lower frequency. So it's 1 year or every 5 years, where we have information on habitat diversity, soil physics or soil characteristics or vegetation. So basically there is this overview. These are just some examples. And what we are doing at the moment is building what we would call a data lake, which is this this access to information, to eLTER legacy data. This is done in the frame of the eLTER Plus project, and this is also where we, for easier access to information, for example, we are using this UDAT B2Drop service as an intermediate data store for data collection and data processing, and also, for example, Time Moore from UFZ is now working with this data. That's not the end. There is we are also working on the harmonization of information, and that's I think would be the main link to the BioDT project. This is done within the science cases of the eLTER Plus project to create what we call pilot data products. So they are, they are screening the data across the different sites, harmonizing them in terms of spatial temporal resolution, but also in terms of observation. And this will be published through the central services like this eLTER Digital Asset Registry, and stored in the eLTER Central Data Node. And that will be, I think, most probably this relevant data sets for BioDT. At the moment, we are envisaging a file-based data provision, which is on this, what we would call this level 2, which are integrated and harmonized data sets across the different sites, which are then accessible through the central services. In the eLTER Plus project, we are working currently on this piloting, and that also will be this main interaction, I think, with the BioDT. Yeah, that was a short overview on what the LTRI is about and where we are going in future. If there are any questions, I'm open.
Yeah, thank you, Johannes. Are there any questions? Yes, Sharif.
Yes, it's a more of a technical question. I was curious about the CDN and the data lake part. Did you go through any sort of exploration of different solutions available currently, or because there are like a lot of different data lake software, but more recently people are kind of moving into what's called the lakehouse architecture, which is like bringing in data warehouse and data lake together.
Yeah.
So something within Naturalis we're also looking into, not for DiSSCo, but other projects as well. In particularly for a lot of analytical work, not for SQL type query, but more on like Spark and other types. So I was just curious, have you done some sort of comparisons or tested with tools?
Yeah, at the moment, the colleagues from so that's the part for UK CH, they are working on that. So they tested different types of solutions at the moment. There is one of the issues. There is no final decision. We thought about for having the SOS and sensor things APIs for the observation data. We looked into a more SQL type collection of information. In the end, it will be, I think, a combination of different solutions depending on the type of data. But there is no final decision on that at the moment, and UKCH is looking into that. That is also why, for example, at the moment we are also looking at services provided, for example, from UDAT, is to evaluate, for example, if that could be used for parts of the data provision, for example, also to publish data snapshots and like that. But it's still an ongoing issue. So
Okay.
there's no final decision at the moment.
Are there any other short questions? If not at this moment, then I would suggest that, because this is a virtual meeting, that we have a 1 minute short stretching break to stand up, and then to keep us refreshed, and we continue at, let's say, 7 past with GBIF. So I don't have a second, how do you call, timer, but I think we can start getting back. I see Marcella's slides already. So unfortunately, Doug Anderson was sick or not feeling well today. So Marcella Orwick Rydmark kindly took over to present GBIF. So please, Marcella.
All right, so thank you, everybody. So today I'm presenting on behalf of GBIF Norway, so the Global Biodiversity Information Facility. Dimitri Schugel should be here, I'm hoping, in the next 10 minutes or so. So he's a part of the Secretariat in Denmark, and he'll be helping out with the questions after this presentation. So firstly, GBIF is a fair and operational infrastructure. So it's an intergovernmental network and research infrastructure that provides anyone, anywhere, free and open access to data about all types of life on Earth. There's a voluntary collaboration with quite a number of participant nodes and a secretariat that is placed in Copenhagen, Denmark, oversees the whole project. So a bit about the participation. We have 40 voting participants in GBIF, 24 associate countries, 43 other associates, and nearly 2,000 data publishers worldwide. And right here is a map where in green we have the most participation, in blue is where we have moderate participation, and white is where we still need to be improving our presence, I would say, in terms of having a global biodiversity and showing all of this data here within GBIF. So just a little bit about the numbers. So these are actual numbers that we just got yesterday. So there are about 2 billion species occurrence records now in GBIF with 64 country participants, 43 organizational participants, 76,000 data sets, about 2,000 publishers of GBIF data, 24 billion, nearly 24 billion downloads per month, and nearly 8,000 papers now that are using the data from GBIF. So the different biodiversity data types that we can take in. So GBIF, as I've just was previously mentioning, has a fairly strong global presence and as well can take in quite a number of different data types. So we take in, for instance, digitized specimens from museums, quite a bit of what Sharif discussed already. Observation data from both scientists and from citizen scientists is extremely important. Literature data, so literature data from both the present and the past. Remote sensing data from different organizations, from academics, from industry, and as well environmental data. And the environmental data is a little bit newer. And here, just because this briefly came up, with the environmental data, there is a massive amount of metadata that also goes along with this, and which also must be digitized and made available with the eDNA data so you can actually make sense of it. So this is all done with the common standards. So the Darwin core, and then there's the data publishing and indexing, which makes the data discoverable and usable. So ensuring that it fits the FAIR principles. So the data richness levels that are supported by GBIF. So the data richness can be fairly light or quite heavy, actually. So we have data sets with the metadata available. We can even just take species checklists. There is occurrence only data, and there's also sampling event data. So depending upon the type of data, there's a different level of richness that can be associated to it and can be found on the GBIF website. And as well, there's quite a bit of species occurrence with multimedia evidence. And so right now we have 122 million records with taxonomically identified images. So this includes nearly 1 million audio files and about nearly 4,000 video files. So this includes about 48 million different specimens with the multimedia and 69 million human different observations. As well, as we were just mentioning, for the sources of data are the natural history specimens that we've been discussing. So digitized specimens. So here are two examples that can actually be found on GBIF from the Mycology Herbarium here in Oslo, and also from the Vascular Plant Herbarium at NTNU. Both of these are within Norway. And so these data right now, they can tell you quite a bit about it, but what I can also say at this point is that quite a bit more of the metadata and the technical data that needs to be associated to these sorts of collections probably still needs to be made machine findable and also digitized. As well, observation data from the past. So I was talking about old data as well. So here are two different examples of literature from the 19th century on biodiversity observations that were made that can also be found in GBIF. And as well, extremely important for us are observations from citizens. So eBird, iNaturalis is international crowdsourcing portals. And here in Norway, where GBIF has quite a presence, then we also have Artsdatabanken, which is a national crowdsourcing portal. And all of these different portals can be directly linked to GBIF for the final publishing of the data. And finally, as I was briefly just mentioning, genomic data and eDNA. And so this is growing very, very rapidly within GBIF and within, I think, the other research consortiums as well. And what I should mention here is that it's fairly recent, is that there has been a crowdsourced book now available on how to publish this sort of data. Right now, it's not terribly well standardized. And for instance, a lot of the metadata that needs to go along with it is not fully standardized yet. And so this is just a cookbook that GBIF has now made available on how to try and standardize and how to publish these sorts of data. And now onto the data coverage from GBIF. So as I've previously mentioned, we have pretty strong data coverage. Where in white are the areas that we still need to do better. But for instance, in the US, in Northern Europe, in Australia, and in Southern Africa and South America, we have a strong presence. And so I think a lot of people already know this, but there is a fairly strong taxonomic bias towards birds and against insects and small organisms when you think about their relative biomass versus the data and the literature that's available on them. And this is also very apparent within GBIF data, where we have an occurrence excesses, for instance, towards birds, but we have occurrence shortfalls when we're talking about insects and smaller organisms. So this is something that we actually do want to still try and rectify in the future with GBIF. And then data trends. So the species occurrence records of GBIF over time. So these occurrences are in the millions. And again, they are very high for animals, a bit less for plants, and then others, for instance, insects and small microorganisms, quite a bit less. And then as well, the number of species having occurrence records that are accessible through GBIF over time. So the number of species here is in the thousands. And again, you can see a similar trend. And we would like to note that within animals, it's a lot of species occurrence for birds that we have in GBIF. So birds are very, very well represented within GBIF data. And now a note on this, and this is quite related especially to Sharif's talks on museum specimens and their digitization. So natural history museum collections probably have something between 1.2 and 3 billion specimens worldwide. And these are incredible sources for research and provide snapshots through time of our world's biodiversity. There's a lot of challenges working with these data. Oftentimes you don't have the associated metadata that you would like to have with it. And obviously there are oftentimes not very many replicates as you would like to have. But still, even with these challenges, people are working quite hard to work with them. Now, GBIF has about 2.2 billion records overall, which includes about 200 million specimens from museums. So right now, we think that there's about 10% coverage of all of these museum collections worldwide in terms of getting them digitized. So we still have a pretty long ways to go. Now, Scandinavia and Norway also, I will say, is actually doing really, really well within the digitization of museum collections. And so this is just a snapshot of the museum collections at the Natural History Museum where I work. And so we have approximately 6 million specimens total. And so you can see that we've actually done a pretty good job with our digitization, where we have 83% of our botanical collection digitized, only 20% of the zoological, so we need to do better with that. But then again, 63% of our geological collections, which are less interesting for BioDT, but are overall extremely interesting for the natural history museums and understanding world's diversity in general, as well as about 50% of our paleontological collections. So this is a big effort on our part to be pushing this, and this is a continued thing that we're working quite hard on at the museum. Now I was also just talking about some of these challenges. So one way that of digitizing these collections. So GBIF in the past has actually had a crowdsourcing platform, something called Dugnad. So Dugnad is a term within the Scandinavian language that has to do with bringing together people for the societal good. So we've actually had a website where people can where we have these snapshots of metadata, and people have actually helped us with transcription and digitization of all of these labels, which has been massively helpful and actually increasing the speed at which we're getting these things digitized. GBIF data publishing. So it's not that difficult to publish your data with GBIF, but there are some things, of course, that are necessary. So the step one is the digitization of the data, the collections, the herbaria, whatever multimedia file that you already have, which is, of course, digitized, but that is a first step. Step two is the registering for endorsement within GBIF. Step 3, and highly critical, is conversion so that it's compatible with the Darwin core format. And step 4 is publishing from the national GBIF node. Now, alternatives for this can be publishing from the regional GBIF cloud data repository, as well as many citizen science platforms already publish data directly in GBIF. So iNaturalis, as I mentioned, and for instance, here in Norway, Artsdatabanken already work quite closely with GBIF to ensure that they get their work published within it. And now a bit on the data use. So GBIF has a pretty strong policy links with their work. So with the Convention on Biological Diversities, as well within UN sustainability goals. And so here we have one snapshot of how GBIF data in Norway is used within the Convention on Biological Diversity. And so these are some policy goals with the AKE targeted supported biodiversity indicators, where they have actually come back to us telling us how GBIF Norway is doing in supporting their goals and supporting biodiversity. So GBIF Norway is seen as supporting, for instance, trends in invasive alien species through global registration of introduced and invasive species. GBIF Norway can support the Species Protection Index and Protected Area Representative Indices. It supports the comprehensiveness of conservation of socioeconomically and culturally valuable species, including the Agrobiodiversity Index, as well as the Crop Wild Relative Index. And finally, GBIF Norway is seen as supporting the growth in species occurrence records that are made accessible. And that one is not too surprising, but because that is within GBIF's overall mandate is to have these species occurrence data. But that's also very closely tied to these international policies that we're also trying to support. And as well, besides the policy links, GBIF is quite important for supporting research and sustainable development. So for instance, within conservation, protected areas, threatened species, invasive species risk, within food security, crop wild relatives, in situ and ex situ conservation of genetic diversity, fisheries planning, the data is used there. Within climate change, modeling impacts on species ranges, adaptation strategies, mitigation benefits and risks, and as well as in human health. So disease risk based upon occurrence of vectors, hosts, reservoirs, medicinal plants, hazards, and for instance, as snake bites. And so within BioDT, we're helping out with at least 3 of the different user cases related to this conservation within food security and with human health. So there's a lot of data within GBIF that is very important for the BioDT project specifically. And here, just briefly, upon the peer-reviewed publications that are using the GBIF-mediated data. So we're doing quite well with this, upward trends. And so currently, there are more than 3 papers per day that are published using GBIF data. There's been well, we're not quite done with 2022 yet. I'm not exactly certain when this was published, but the trends are going exactly when we were hoping. So GBIF was established in the early 2000s, I think 2003, and we've grown significantly in our presence also within the academic community, as well as our links to the policy that we've already discussed. And just very briefly, because this is actually quite important, is the data citations within GBIF. It's very easy to cite data from GBIF. And this is extremely important actually when we're trying to get people to publish data, especially getting academics to publish data on GBIF. Making it easy to cite their data and for them to get credited is actually extremely important for us. So one can simply download the data from GBIF. They receive a recommended citation with a DOI, and this DOI is cited within the published research or other work. it's really actually hard for me to emphasize enough, especially for the academic community, how important this is to make GBIF data and data that we upload easily citable. And as well, with this findable, accessible, interoperable, and reusable, these FAIR principles that we discuss. FAIR data is also about machine readable data. So research and museums having their data on the website in a way that it is reusable typically means machine readable. And so we've already had quite some discussions about this within the BioDT user cases as well, is making certain that the data that we want to use within the user cases, for instance, is FAIR data and that it is machine readable, because otherwise within BioDT, it's going to be hard to use these data. And this was actually something that came up with within the last user case workshop, where we had a fair number of questions about whether all the data that we plan to be using within BioDT was FAIR data. And this is something that we do need to try to hopefully manage. And so just very briefly, a new tool within GBIF is something called Bionomia. So Bionomia is looking to better link, I should say, natural history museums to the world's collections. So what we're trying to do here, for instance, is make certain that GBIF data is just simply better integrated with some of these other websites. And as well, and interesting, when you're thinking about a lot of these museum data, oftentimes we don't know the primary authors within the data that's actually published online. You're talking about people from the 19th century oftentimes. And so Bionomia actually has a real case here of trying to ensure that the original scientist that actually collected the specimen can also be found within the metadata that is collected within any sort of specimen that has data within GBIF. And so that is the GBIF presentation, and I would like to thank you. So this is Dag, who is meant to have been here today, but unfortunately is not feeling so well. And so I am hoping that Dimitri is soon available to be answering any questions.
Yes, thank you.
Here, yes.
And
Great.
On the call. Jan Dick, you had the question in the chat. Do you want to ask it out loud as well?
Good morning, Marcella. That was an excellent overview. I was particularly interested in when you spoke about the audio files. I have recently been invited to take on the ecosystem service use case, and I'm still trying to identify what I will do. So I wondered if you could tell us something about those files. Are they all biodiversity focused? Are they the soundscape in general, or?
So Dimitri, can you answer a little bit on that?
Yes, I can, yes. If we have time, Terhi, do we have time for questions now? Yes,
yes,
good.
Yes, this is the time for questions, yeah.
Thanks, Marcella, for presenting brilliantly on GBIF. I can see that I should stop doing this and should always ask you. On the question of the audio files, I think if I'm allowed, I would like to share screen very quickly, just to illustrate that some of these 2 billion plus records that Marcella was presenting about actually have different kind of multimedia. Most of it is images, and most typically it is citizen science observations. You take your smartphone out from the pocket, take some snapshots, get them identified, and if people agree on the name online, it gets research grade and becomes a record discoverable in GBIF, among other biodiversity types. In the more advanced cases, the record of the species presence or maybe even the species movement track can be accompanied by video or audio. And no, it's not all birds. I was looking when I saw your question in the chat, and I didn't have to go very deep to find gecko, which apparently is a reptile, right? I'm in invertebrate zoology, but observed in Bali, and I'm. And so the confirmation of this being gecko is supported not only by somebody's opinion or pictures, but also with the sound. And similar for videos. So to find these things, if you go occurrences, I hope you can see what I'm doing in front of you. So you just go to gbif.org, click occurrences, and here you can select media type. You can see there are 122 images. Only 3,000 records would have any video with this. Of course, there is a storage issue, but almost 1 million records with audio files. And if you click that, you can and go to metrics or taxonomy, you can see the breakdown by the different taxa. Yes, you're right. Most of it would be birds, but I think there is a lot of insect audio files and so on. I can continue a long time, but I think there might be other questions here. I hope I answered it, Jen.
You did, and I'm fascinated to know what the sound file for the fungi is, but I will follow that up. Thank you.
I can tell you how it's done, yes, later.
Yeah, thanks. Are there any other questions? Please use the raise of hand function. Do you have a question to yourself, Dimitri?
I just wanted to add a little bit for those who are new to the GBIF in BioDT, just to clarify that when we were writing the project and now when we are running the project, it was realized that we'll have a choice of depth and breadth of coverage of our data sources. So GBIF represents the broad and shallow. So people digitize and publish biodiversity records from all kinds of sources across the world. And we can provide centralized access to citizen science and museum records, let's say for birds, if you want to do a continental level modeling. But we were working together with other research infrastructures, specifically when we discussed the difference between use case building based on eLTER versus use case building based on GBIF, we realized that eLTER is on the other end of the spectrum, representing very valuable long time series with documented sampling effort, quantitative data. So eLTER is deep, GBIF is broad, and across 8 use cases we'll have an opportunity to benefit from both, I hope, and we'll probably learn that the contributions from DiSSCo and LifeWatch, they're more in the infrastructure to infrastructure interactions rather than direct content provision. I hope I'm not telling something that you disagree with, but okay.
No, I think that was a relevant addition. Johannes. Open. Yeah.
They just recognized that I have muted myself. I have one question, and thanks Marcella and Dimitri for the presentation of GBIF. One point we also discussed quite a lot is, you know, this interlinkage between eLTER and also how we, for example, could link up with GBIF in both directions. So contributing data, but also providing information on these observed areas or on this site context. One point we discussed there are these environmental observations, so these abiotic observations, which I never really clearly got, because up till now I always thought this more is this environmental conditions you record during the observation itself. Is this, or is there any idea, for example, to combine that with a broader scope? Because, for example, as we have it in eLTER, basically you have the biodiversity observation on the single plots, but you also have this kind of environmental observations which accompany this, you know, from deposition, soil processes, and so on, which are normally near these locations. So maybe you can expand a bit on this environmental part in this Darwin Core extended. So how that is done, because as far as I understood, that's linked to the sampling event, which is a quite important extension. I think also in the perspective of eLTER. So maybe you can have some words on that.
Shall I continue, Marcella? Can we have still an answer here? We have time?
You can have.
Thanks, Johannes. This is very, very good point, and I think it's very important to clarify that when we publish data such as eLTERs, and if you decide to resurface it to additional data discovery avenues, for instance, if you come to the decision to publish biodiversity data also to make it discoverable through GBIF, it's a lossy process, and people who have deep interests for the full analysis of this data, they might not be happy with seeing only biodiversity data, and then they would be wondering where is soil chemistry, where is temperature, where is precipitation. We have gone through such discussions with NEON. I'm sharing my screen now with you. I hope you can see it. Yep. And they found value, which is reflected in this 157 citation of their 40 so far data sets to show only biodiversity component. But then with this data, you can click deeper. You can go to the NEON data portal and find the data in its full complexity. So basically, GBIF starts to work for the network like NEON, similarly as Google works for the general internet search. Mm-hmm. When you find your result on Google, you don't see it in its fullness, but you know where to find it at source. And we have gone through a similar process with molecular data partners, Magnify, GeneBank, Barcode of Life. Of course, these systems, they have their own portals, own audiences, or own ways to discover the data. But resurfacing through GBIF brings, first of all, expansion of the audience, but specifically for the BioDT context, this data become available through the standard API. Mm-hmm. So you can plug it straight to the use case. And because it is done with credit, you never lose the original attribution. So you see, like on my screen now, you can see it says it's publishing by the National Ecological Observation Network. It's hosted by the USGS and so on. So we never show it as a GBIF's data. There is no such thing as GBIF discoverable data. And as Marcella explained, DOI preserves the origin. So if something like that is of value, we can repeat the trick or experiment at least with something small and safe and see if it brings something helpful.
Okay, thanks.
Thank you. Then I see still Christos' hand up, and then we can maybe use that as a bridge for LifeWatch presentation as well.
Thank you, Takri. There is a noise at the airport right now. That's the reason I couldn't join from the start. Can you hear me better now?
Yes, we can.
All right. Then I will go back to the point that Dmitry said, before about the technology that is available right now and allows us to discover combined information from different sources, and also find the provenance of that, and through this combination we can and can use GBIF's data. Sorry, I lost you.
Yeah, we lost you also for a little while, but the beginning we heard.
Yeah, I was saying that the technology, my colleague will talk about that in a minute, is capable of bringing combined resources, combined data from different resources to the researcher. Therefore, the exploration and also the exploitation of the data of GBIF can be more wide and can engage more disciplines. And that's fantastic because we have already started in bicycle, we have already started in near future. So all these developments will flow technology and capability in the biodiversity DT. And one last point is, when we talk about, biodiversity resources, we tend, to forget about the molecular ones, all right? Those that Elixir provides, ENA provides, and other resources. We should think about, how to engage those resources as well in our hypothesis. Thank you.
Thank you, Christos. And then I think we move on and continue with the LifeWatch presentation. Christina Huertas from LifeWatch ERIC has promised to give this presentation. So please, Christina.
Good morning. I'm going to present LifeWatch ERIC, which is the e-science and technology European infrastructure for biodiversity and ecosystem research. What we are, LifeWatch is an ERIC that provides e-science research facilities to scientists working on biodiversity and ecosystem structure and functions, and the services they offer to mankind in order to support society in addressing key planetary challenges like climate change. Our vision is to become the research infrastructure providing access to the world's biodiversity content, services, and communities in one click. As you see, our mission is very large. It is to accelerate the research efforts of the scientific community by delivering the European state-of-the-art infrastructure on biodiversity and ecosystem research, a digital twin. We are a distributed research infrastructure. We have 3 common facilities in 3 different countries. In Spain, we have the statutory seat office and the ICT core. In the Netherlands, we have the virtual laboratories and innovation center. And in Italy, the service center. We also have distributed centers in Portugal, Belgium, Slovenia, Bulgaria, and Greece. We offer a prototype in TRL 6 with different research resources like data sets, services, virtual research environments, workflows, training resources, and research sites. Our core services are the Eco Portal, which is a semantic repository, the metadata catalog, the Tesseract that includes Jupyter Notebook as a services for virtual research environment development, and the data block for data and other resources, discovery, management, provenance, and monitoring. This is how a workflow in Tesseract looks like. It can be a bit complex. And this is a live block development. As you see, it has 3 main units. The first one is for the engagement of the research infrastructures. Then we have here the main technology that integrates the resources of the live block. And each provider can have their own live block node. And here you see the 3rd unit, the semantic database. And through the semantic search, the users can have access to all in the integrated resources and make complex questions. We also offer training and citizen science. For example, LifeWatch is involved in different master's and PhD programs, and also in summer schools. This is our new conceptual architecture. It goes all the way from the data lake to the interpretation of the results. As you see, it has several layers. What we offer innovation. We offer access to multiple data and services resources in one click. For example, access to biological and environmental data at the same time. We provide workflows dedicated to specific lines of research with customized data and services. We enable the users to develop her or his own workflow depending on the working hypothesis and the content and services available on the fly. We serve individual researchers or their communities from multiple disciplines or domains by providing multiple workflows that can test the same hypotheses in parallel and then compare the results. We have identified 11 stakeholders in the public sector and 3 groups in the private sector. In brief, our contribution to BioDT has 3 components. The first one, the fair, compliant data. The second, the reproducible analytics. And the third, mobilized communities. Summing up, our areas of focus is the biodiversity and ecosystem research. We offer research resource of all possible levels of the biological organization and scales of observation. As we previously said, we have identified 11 stakeholder user groups from the private sector and 3 from the private sector. We offer 115 services to be used either individually or orchestrated into workflows, theories, or thematic services. Regarding the data architecture that we have previously seen, we have the Eco Portal, that is the semantic repository, the metadata catalog, and the LifeLog, that is the integration of data or any other type of research resources, such as services, publications, database and repositories, observatories, individuals and communities, and publications from every research infrastructure or global aggregator. PLATS is semantic database, which make it possible for the user to ask very complex questions and receive replies. For example, give me the species and the specimens collected by Alexander Agates in North America. We have already talked about the metadata, and regarding the tools to access, we have web services for discovery, management, monitoring, evaluation, and attribution of data or any other research resources. LifeWatch ERIC scheme has been already published, and it contains different components like deriving code, semantic, metadata catalog. For the time being, all resources are openly and freely accessed. And finally, LifeWatch ERIC has no collections in its infrastructures. So thank you, and if you have any questions, I will be happy to take it, or our CEO, Christos Arvanitidis, who is also in the workshop.
Thank you, Christina. So we have plenty of time for questions. So you can raise your hand. Yes, Thomas.
Okay. I would like to ask about the difference between the Tesseract and the LifeBlock, because both you showed them really quickly, and both had these some graphical user interface connecting blocks, but I'm not sure what's the difference.
Okay, I will pass the question to the CEO, because now that is between us. Christos?
Thank you, Christina. I hope that my connection is better now. Tesseract, which includes Jupyter's Notebook as a service, is a tool to create and implement the virtual research environments. That is, the user can make his or her own workflows by just linking one service after the other under the assumption that the output, from one service is used as an input from the next one, and so on. So there are many systems like Tesseract, of course, and as I said, it includes Jupyter Notebooks as a service. About the LifeBlock, this is the implementation of blockchain technology in the environmental sciences, and that allows the aggregators, the research infrastructures, and many other interested parties to engage with LifeBlock and then of course to create, their own nodes. And through this, the technology allows all the users to know who is using what resource, when, and how, and also allows, to provide the provenance of the resources. And as I said, before, this is the technology that allows, multiple, access to multiple resources at the same time. It brings all the metadata at the same time. And also because of the semantics database, the user can make very complex questions and get replies from the system in terms of resources, in terms of existing resources from the engaging communities. So there is a big difference between them. Did I answer, Thomas?
Yes, yes, thank you very much.
Okay, I think Klaus, please.
Yeah, probably related question. It is not, or I'm quite curious how these things work together. I mean, I knew EcoPortal a little bit, and I knew that you store already mappings there, which is also, of course, one of the targets of WP5. Are these, is the provenance, for example, for the mappings, is that stuff also stored in the blockchain, or how these components working together? It sounds a little bit like the blockchain is providing a kind of middleware where all the provenance data and so goes in and has probably the data models for the mappings, or are these unrelated services?
Klaus, I'm not sure if I understand the question. I'm a biologist.
Okay, sorry. How are EcoPortal and LifeWatch also connected together somehow, or are these separate applications?
Yes, they are. Yes, they are. EcoPortal is our semantics repository.
Yeah.
That means for our resources, we need to know what is connected with what. The machine needs to know what is connected with that and how. Okay. So that's also, included in the functioning of
Okay.
LifeWatch as well. And it's not only about our repository, because if other resources come with their own repositories, that can be used as well. That's why they developed, the semantics database.
Okay, thank you.
And as Carol is mentioning in the chat, that workflow might mean different things in different contexts. So could you still clarify what workflows means in LifeWatch?
I did a moment ago. For us is, linking resources and developing complex environments where the user can test, a hypothesis with one click from top to bottom. And also in some cases for which we have already the prototype, the user can test the same hypothesis through different approaches. For example, the conventional community analysis and the metabarcoding community analysis. And then she or he can compare, even the results. That's how we perceive the workflow working with our resources, or the resources we have access to. But I'm sure that Carol has a lot more, to say about workflows, because it's her, specialty.
Yeah, so please, Carol.
Yes, I mean, throughout the entire workshop this morning, workflows have been used in every single presentation with slightly different understandings and nuances about what workflows mean. In LifeWatch, it's a kind of a process, a scientific process, which would also include a human in the loop. It includes, which is why there's a big emphasis on Jupyter Notebooks, although Jupyter Notebooks actually don't constrain you to go one step after the other. They actually can allow you to go wherever you want. So they're an interesting sense of workflow in that sense. They're only workflow by convention, not by actual implementation. Whereas other folks are using computational pipelines that are actually using Spark architectures, or are using computational instances. So Galaxy is used, for example, in the DiSSCo project for the specimen data refinery. So I think it's a meta question I'm actually raising here, which is for us to be, as we are working on how do we harmonize our data and how do we harmonize the terminology. We also have a kind of understanding of the spectrum of workflows across the research infrastructures and the different kinds of technologies that we're using then, how we can also bring those together. So I think it's quite interesting. I'm interested in all of these, of course, because I run the Workflow Hub, which doesn't care, it just has all of them. But it's an interesting, I think it's a useful thing for us to do in the project, because it's something that will get quite confusing if we're not careful, if we really mean a business process type workflow, as opposed to something that's doing job scheduling on a cluster. These are all workflows.
Yeah, it is very true that agreeing on use terms is important to work well together. Jesse.
Yes, just continuing from that point, there is a BioDT key terms page on the wiki, which I'm going to place in the chat, because currently there is a workflow definition in there, but it's very narrowly focused. So I agree that it's important to have these different definitions outlined. So I could ask that the RI representatives also add their own definitions of workflows there. Thank you.
We did that exercise from LifeWatch, no? Haven't we?
Yes, but others as well.
Yeah, of course. One more point, for Carol, even, in Tesseract, the services and the resources are free to be used. So the user is the one who defines how the workflow is being built freely.
Yes, I don't disagree with you there. It's just that there's across all the different research infrastructures, there is a sort of wide variety of workflow stakeholders, workflow users who are effectively users of pre-cooked workflows, workflow users who are actually developers of their own workflows or adapters of other workflows. So there's a big a kind of range of different kinds of stakeholders and different kinds of capabilities and different kinds of workflow systems. We're currently up to 324 different workflow systems that I'm aware of. So there's quite a busy landscape when it comes to workflows.
Thank you. Are there any other questions for LifeWatch or other RI presentations or comments? I don't see any raise of hands. So then we will have about 10 minutes break and then continue with the panel discussion. So let's come back 7 past. I will stop the recording and So welcome back. This second part of the Research Infrastructures workshop will be a panel discussion with the 4 research infrastructures, and Jesse Harrison from the CSC will be moderating the discussion. So please, Jesse.
Okay, so yes, we have 4 speakers in the panel discussion today, and I'm happy we have someone from each of the RIs. So we have Christos Armanitidis from LifeWatch, Ute Skiba from eLTER, Emily Beltien from DiSSCo, and also Eric Kuus from GBIF. So really what this panel discussion is about, well, in BioDT project, we of course want to design DTs that serve the needs of users. So that's one of the main requirements. We want to create things that are actually useful and make sense from that viewpoint. So the focus is in this panel discussion discussing the user's needs and priorities when it comes to interacting with the different research infrastructures, and also in relation to the BioDT project. And we could start with some short introductions from each. So maybe we could try Christos first. or if Christos is working on audio, then maybe Ute.
Yeah, hi. Yeah, I'm a biogeochemist at the Centre for Ecology and Hydrology in the UK. And my focus mainly is on greenhouse gases. So relevant to this session today is that I am leading the work package 8 in the eLTER Plus project. And the eLTER Plus project is looking, and work package 8 is trying to see how we can integrate data, say on something like extreme events, for instance, a drought event. How can we sort of harmonize or sort of combine all the data we have from the different disciplines? And the disciplines we are studying at the moment is biodiversity, biogeochemistry, and then the water-food nexus and socioecology. And so far we have been looking for doing this, looking at the sort of eLTER, the DIMES data, and it's really difficult to sort of harmonize all these 4 disciplines and all together sort of provide data for a drought event. So there are data gaps here, and obviously we also sort of just found from the discussions earlier today that the data gaps are everywhere. So that's all I think I need to say.
Thanks, Ute. How about Emily next?
Hello, everyone. My name is Emily. I'm here for the DiSSCo research infrastructure. So I actually bring 2 types of users, I would say. So firstly, in my current position at INBO, I represent the user type of a collection manager, and more specifically for hidden collections. So that means collections that still have to catch up a little bit with the bigger musea. And on the other hand, I think this is more relevant for you, would be my user type as a researcher. So I used to be a PhD student working on threatened tree species. So I have some background in plant systematics, biogeography, conservation genetics, and a little bit in species distribution modeling. So I think for the introduction of me, that's it.
Thanks, Emily. I wonder if Christos has audio yet.
Yes, I do. Do you copy?
Yes, thank you.
Perfect, thanks. Christos Arvanitidis, CEO, LifeWatch ERIC, Seville, Spain. I started as a taxonomist and polychaete taxonomist and biogeographer. Then I moved towards the community analysis and finally biodiversity research. And my special interest is to compare patterns of information from multiple levels of the biological organization and scales of observation. But lately, I have been enchanted with the cross-domain research, especially in the context of EOSC future project. So I think that's all.
Thank you. Eric.
Hiya, folks. Name's Eric. I'm a third-year PhD student at the University of Aarhus in Denmark. We'll be joining the BioDT project relatively soon, early next year. And my user base is very much in the realm of quantitative ecology. So I use geopositional data as found in GBIF, combine that with climatological data as found in other data sets to then understand how we can infer ecological interactions and networks from those geopositional data and how we can use these distributions to then project what's going to happen to our environment in the future. However, that's very data intense. And so integrating data sets with one another is very near and dear to my heart.
Thanks, Eric, and also welcome to BioDT project, of course. So we see that we have people from very different backgrounds here, many of them very relevant, of course, to BioDT project. So big data is maybe one, of course, but then ecosystem connectivity, biogeochemistry, these will all come into play, of course. But we could maybe discuss quickly also the scientific problems that you've been working on. So, okay, we hear a little bit of information in the introductions on these, but I'm interested especially in which scientific problems you have been working on in relation to the RIs or using the RIs to solve these. So, whichever of you would like to first comment, maybe I can pick one of you. So let's start with Emily, maybe.
Yeah, so I'm going to talk a little bit hypothetically here because I used to be a PhD student. So if the DiSSCo RI would be running at the time, I think the main thing I would use the DiSSCo research infrastructure for would have been to access herbarium sheets, because that was the main the main source of my data for making the data set for conservation genetic studies. And so not only just the record data, but also for the loans, right? So Sharif talked about it. So the main portal that you would use, or the main interaction, would be with Elvis. And then you could query for the different records. And then also, I think you would win a lot of time instead of emailing different institutions for loans or information that's missing. You would have like a one-stop shop for your information and for the whole transaction between providers of collections and users. So yes, that's my answer.
Okay, we can focus also on the exact scientific problems. So we will come to ways to interact with the RIs quite soon, but we would like to go next. Maybe you can raise your hand. So okay, Eric, please.
Cool, I'll go for it. Thank you. I struggle to come up with a project that I've worked on recently that hasn't involved GBIF in some sort of capacity. So for one of my PhD projects right now, I am contrasting the effects of ecological network projections of biodiversity and ecosystem functioning when identifying primary extinctions and then tracking secondary extinctions by different extinction proxies. For that, I'm using the IUCN Red List criteria, where species haven't been classified by the official IUCN Red List. I then use geopositional data from GBIF to calculate that with a few algorithms. But I also identify local climate optima and local climate-driven extinction proxies by first establishing climatic niches from GBIF and then contrasting that to climate projections in certain locations, given the locations where networks have been established. So I am using the wealth of information in GBIF, combining it with a lot of other things to then understand what's likely going to go extinct. And spoiler alert, it doesn't look good.
Thanks, Eric. But maybe would they be happy to go next?
Yeah, just listening to the talks this morning, or the topics, they're all sort of biodiversity related, obviously. But is it not possible, and all of them are slightly different, but is it not possible to sort of harmonize them and sort of have one large database for biodiversity?
So later on, I think we will talk about cross linkages also between different research infrastructures. But what you raise is a very good question, I think. But at the moment, I guess, so you're working with eLTER,
yeah.
And I think you gave us a good description of the different types of topics that you've been working on, but is there something that you would still like to elaborate on?
Okay, yeah, so basically we want to sort of integrate the different disciplines, which is biogeochemistry. So there you would, for example, look at climate change issues, and sort of nitrogen deposition to the soil, and what's the impact on that on say the plants, sort of certain plants. If there's too much nitrogen, they probably wouldn't like to grow in certain environments. So that's one, and we want to harmonize that also. What happens, for example, for the nitrogen when it goes into the river at the same site? What are the implications there? And what are the implications? Yeah, that was the biodiversity, and the biochemistry would be sort of greenhouse gases. And what is the impact on the socio-ecology? So we want to sort of look at an ecosystem and see what are the issues if you have an event like a sort of nitrogen input to the ecosystems. So that's what we're trying to deal with, and we've got lots and lots of data in eLTER, really good, excellent data, long-term data, and I mean for our research we also need to have long-term data, but it's very difficult to find a single site even that can serve our needs for the 4 topics. That's a real struggle, and I'm sure you probably in your different GBIF and the other top, the LifeWatch, I'm sure you probably experience the same issues as we do.
Okay, thanks, Ute. Christos, you have your hand up. So.
Yeah, the research is going on in our infrastructure towards 2 main axes. One is the global scale, that is how to test standing hypotheses, either taxonomic or biogeographic or even ecological ones, with the data that are available from our infrastructure or through access to other infrastructures or global aggregators and so on. And the other one is this current socio-ecological systems. We are involved in many European funded projects in this. It is a very promising field, actually a new avenue, because of the different disciplines that they have to sit together and work together. And that creates a lot of challenges, but also a lot of innovation at the same time. And, you know, just to try to reply, to the previous question, I'm not sure, for the time being, if we tried to include all of the biodiversity data in a single database, would that work with the current resources? It would definitely be possible with technology, but also with a very big investment like the one they did, for the molecular data, for the molecular resources, such as, the gene bank and the barcoding infrastructure, et cetera, et cetera. The point is that we have so many different types of data that require, quite a lot of work, and it will require, quite a lot of work in order to create these metadata that are capable for linking all of them in multiple ways, of course. I know that there is a lot of movement right now towards linking data from different resources, and GBIF is a champion on this. They have, came up with this Darwin core standards early on, and they are making, brilliant work, and many, if not all, of the research infrastructures we follow their standards, and that's fantastic. But if you would ask me, that is it possible right now with the resources we have, to include all the data in a single database? I think that that would be, not possible right now. It's a Herculean task.
It's no small feat, to be sure, but what I could ask at this point as well, so we've had introductions from all our speakers, so do any of the audience members have questions for us as well, especially to do with the, let's say, the scientific problems that we've just been introduced to, or the actual fields that everyone is working with. There's a question, at least from Johannes, please.
It's less a question, more a comment. I think it's what Christos just said. It's very variable, and I think this is also one way forward. We are going with the implementation of this S3 research infrastructures on a European scale, because I think one part is to integrate and include everything into a single database, which is really a huge amount of work which needs to be done. It's also a question of governance. I think it's also what is behind GBIF. There is a lot of effort done, for example, bringing together this biodiversity data. And I think it's in the frame also of this research activities, and also BioDT is a very important part with the work package Carol is leading, or also what M3 is trying to do. I think one step forward is really to focus on the research infrastructures, and basically what we try to do within the eLTER research infrastructure is to focus, for example, on core variables, which are called the standard observations or the standard variables. It's also a bit to make the scope a bit more clear, because there is, early on when we presented the LTRI, we have about this 520 sites. And when we started, the story was always we have this 520 sites, and basically you have 800 different ways of doing the same thing. And I think this is really a great step forward in the single domains to bring this harmonization forward. So this will be, I don't think, done in one run, so that by the end of the implementation phase, for example, you have everything ready, but to really walk along the line. And I think there are these two aspects. The one is looking into the future and try to harmonize the things, for example, like ICOS did it, which is a huge task, but it's working towards the future. But for a lot of these research infrastructures, and DiSSCo is, for example, also one of them, and LifeWatch is also addressing this, is also looking back into the past and integrating this legacy data, where you have different issues which needs to be tackled. It's the distribution and diversity of the harmonization of the data as it is provided. It's an issue you have to deal with to get sufficient metadata, because sometimes the observers are already not anymore in the organizations. There are some handwritten protocols and metadata and so on. So there's quite a lot of information to be done. But I think you can deal that by the different domains. And what we have to do in the context also of BioDT and all the other projects is to align the different research infrastructures so that this information flows could be easier be done. The FAIR principles are an important part. The services developed by LifeWatch are an important part. There are also other services in the OSC. And I think that is really the way we should go forward.
Thanks, Johannes. Yes, I think we should also carry on with this discussion outside the panel meetings. Of course, it's a large topic to discuss. And I see a couple of hands from Ute and Eric up, but I think what we could actually do is carry on with this discussion at another time, because I'd like to bring this back to the individual RIs as well. So of course, we are interested in how the different users are interacting with the different RIs in this project. And from a user perspective. So could you please also comment on this, your main ways of interacting with your research infrastructures? And maybe we can start with eLTER. So Ute, please.
I personally don't really interact with the. What do you mean by interacting with the research infrastructures?
So actually using eLTER services, yes.
Yes, okay. I personally don't do this, but certainly the members in Work Package 8 interact quite a lot with these research infrastructures. And if you want to sort of try, the problem also is with research infrastructure. Certainly in the UK, there's an awful lot of, there's not a lot of money available for research infrastructures. And so we, for example, if you want to sort of ask for data from some of our sites, it's a very slow process to get the data because the staff don't really have the time to collect all the samples, and on top of that, digitize them in a spreadsheet to then pass it on to other people. And I'm not sure if that's also the case in other countries. Probably is. But somehow often the information flow is not as good as it could be.
Okay. Any comments from the other RIs or additions?
May I?
Please.
Just a quick comment. It's not only harmonizing and trying to find ways to streamline the data into the analytical framework we developed here for the biodiversity digital twin, but it's also its maintenance, because a digital twin is something that is representing the physical world in silico, and therefore we need to find the mechanisms to maintain the streamlining of data. I mean, even after the end of the project's life. So that's an additional problem, to repeat the same kind of analysis at certain time intervals and then compare the results. And if we are lucky, compare the patterns we have, in silico with the patterns we get from the physical world.
Thanks, Christos. Do you want to comment also on, are you interacting directly with are you actually using LifeWatch services yourself?
Yes, I do.
Yes, I would think. Do you want to elaborate?
No, well, one point, for the data is the discovery of the data from different resources, and this is where we're focusing, right now. Probably there is this technology, there is this prototype that can be used, in biodiversity digital twin. Of course, there are some use cases that they need, to be analyzed. It's one thing, when the scientists talk together and they come up, with a pipeline of work. And it's another thing when this is communicated, to the developers, to the engineers, and they have, their own way, to think and put things, on the paper and then in the machinery. So one very serious challenge we have is the language barrier. And I mean, both between the research infrastructures, but also the communities that they are carrying in the project. And we will have, the opportunity to discuss. There are many meetings already scheduled. But this is, for me, this is the most important thing, to happen, first of all.
Thanks. Any other comments on actually your personal ways of interacting with the RIs? Eric has his hands up.
Yes, I'd love to jump in on that. I, in my work, really focus on programmatic retrieval of data via different coding language interfaces. And Christos just raised a very important point here, which I think is the longevity of our data workflows, which a lot of work has already been done on developing good API frameworks that can efficiently query a ton of data. But seeing how we've talked about creating a very holistic database sometime in the future, or at least linking all of the existing RIs to create a holistic data framework, we will end up with a data structure that is even more complex than what is currently already present. And I think we have so far left a bit of development potential open, not to step on anyone's toes here, in making these programmatic interfaces easy to understand. And as far as I'm concerned, that is a big, big development need for us, not just for BioDT, but also for the bigger impact of what we're doing here outside of BioDT, to create material and workflows that can be understood and used effectively by a layman without specialized training for years on end before they can start using stuff.
And this brings us quite nicely to this topic or question about your specific needs in terms of doing research in the biodiversity field. So this could be topics like improvements that you could see on the usability of the different research infrastructures, questions of data quality, are you dealing with missing data, missing models, so forth. So I think for the next 10 minutes we can talk about this topic. So it's basically open floor. If you would like to comment.
I'd just love to jump right back in because I've got some strong opinions on this stuff.
Yes, please continue.
Sorry, I'll take a step back in just a second here. I actually wanted to comment on this earlier when Ute talked about it. I think, at least in quantitative ecology, we have focused for decades now on space for time substitution approaches. And it is high time that we make use of the information that is contained within time series data. And for that reason, I see huge potential here to combine different research infrastructures and their outputs in getting us to understand not just spatial patterns, but also temporal patterns in biodiversity and linking it to the environment. And on a very personal note, and then I'll be quiet and take a step back, when querying spatial data, this is something that I have run into with quite a few data sets. So I've coded some R packages to clear that on the user end. When querying spatial data, I think we should start realizing that people might not need data in rectangular bounding boxes, but in very complex shape files, and it is not always efficient to query a big rectangular box and then cut it all down on the user end. We're not doing our service a benefit here, and we are definitely doing the users a disservice by bottlenecking downloads. So I think the two big things for me is time series data and efficiency of data retrieval.
Thanks, Eric. And I see Emily would like to comment next, and then we could also take a quick question from the audience, but Emily, please.
Yeah, all I just wanted to say is that I think compared to Eric, I'm a more simple use case here. So I think for me, most researchers, well, everything is about time, right? So you have very little time. Well, it's actually complementing a little on what you said. So it's all about time. So you have a very, well, you have limited time to finish your research question, your research project. So for me, personally, I just want to get the data and I want it to be qualitative, and that's it. And I want to be able in all the different RIs, so also the GBIF, for example, what I want to see is like the quality mark, like, okay, what's the quality of this data? And if I have very little time, I can just filter out the most qualitative data and get on with it and make my first calculations, for example, my first database, personal database. So I think for me, or for the research that I have done, it's really all about efficiency from the user perspective and about speed, I would say. So I don't have a lot of time to do data curation or data wrangling, or as you say. So I think, yeah, in the whole digital twin concepts, you know, I want my digital twins to be ready as soon as possible, I would say.
within 3 years at least, within the lifetime of the project. But,
yes, I agree.
Right, it's a good time to take one or two quick questions from the audience. So especially to do with these topics that were just raised, I actually see that Dimitri has his hand up. So if Dimitri would like to comment, then please.
Yes, in fact, my comments might be obsolete after what Emily and you just said, and I was about to actually make the same point, that there were some very, very good ideas here on how we can connect all the biodiversity data with all non-biodiversity data in the deep, bright future. And also back to the point that Christos made about the cost of such infrastructure. But it is very important to realize, and it must be Yes's line, not mine, that we are in the 3 years long project. So we'll have to somehow combine our vision long term with the brutal reality of the project duration, which actually has less than 3 years left. So we need to be able to demonstrate by the end of the project in the BioDT prototype that these things can be done. They don't have to be comprehensive, but they should be representative enough across 8 use cases to show that modeling can move from standalone exercises which terminate with research publications, which some people cite, others don't, to something standing and dynamic, to the on-demand service that can take real existing data streams which are available here and now, well, let's say within the first year of project, that we can plug into the modeling machinery and try to combine them together so you can, for instance, at the same time model deforestation, agrobiodiversity differences and changes in the water table and the freshwater availability and precipitation. Less forest, more fields. What does it mean for the biodiversity? And what does it mean for pollination, or what does it mean for bird migration? So this kind of things, they may sound very simple, even banal for people who are asking deeper biodiversity question, but I think for the sake of the project mission, we need to remember kind of all these visions that we have now in this very useful discussion, together with like dry needs to do things as promised in the project document. It's a very hard combination, but I think it's fairly typical for project-based work, right? You have to somehow harness your ambition for the time and the limits of the project you have at hand. But I'm sure we are in excellent company, and we have the workshop on the use cases in Potsdam, and also listening to the great comments today. I'm actually very positive and confident that we can build a functional prototype. Thank you.
Thanks, Dimitri. Any comments on the or from the panel members on what Dimitri brought up?
Did we discuss sort of gaps? Like, I mean, Dimitri, you just mentioned the birds in your presentation, that the biodiversity, mostly that birds are studied and not the other stuff. So basically, there's obviously skewed representation of the data available on biodiversity, and the same is for the other disciplines as well. How can we address that, and how can we address the sort of issue with lack of data in some countries and lots of data in other countries?
Thanks, Ute. Any other additions from panel members?
I think it's hard to find 4 different ways of agreeing with what Dimitri just said. But to come to what Ute just said, I think finding data gaps is something that can be approached in a manifold set of ways, and I think solving that is probably among the things that Dimitri just brought up, something that we can start now, but will take decades probably to fix.
Thanks.
Yeah, Sharif has his hand on a while.
Let's take Sharif's comment quite soon. Emily, if you want to comment still.
Me first?
Yes, please, and then we do Sharif after.
I just wanted to say that in the question of Ute also of how to get the missing data, I think initiative like DiSSCo is very helpful because it's made a little bit on a lower scale than, for example, GBIF that tries to combine all the biodiversity data, but that you really, focus on a certain community, which in our case is a collection community. And as in the presentation of, I'm sorry, I forgot her name, but in the presentation of GBIF, as it was given that only 10% of the collection data, which is historic data, is present. And so the community of the collection managers and the museum and all those hidden collections that, for example, are also present here in my institute. So in a more targeted approach, I think you can get different communities, their data, in the mix. So I think it's not impossible, but it will not be in 3 years' time, of course. So that ambition is another question, I would say.
Thank you. We can take a quick question from Sharif for a comment.
Yeah, it was similar to the conversation about lack of data and missing data. I was curious, like as a researcher, for example, if you know GBIF, you'll go to GBIF first. If you know eLTER, LifeWatch, DiSSCo, you'll go there first. But and there was a comment about, do you include your social data in your model because you don't know where to find it? What would, as a service provider, we can help researchers to basically let them know where to go? I mean, is there something we should do, or is there something that more of the training for the users? For example, like if you're reading a paper, there's a link to data, and then you click on that, and you probably need to hop on a few places to go and get the data. And sometimes some of those links might not work, sometimes they work. But is there something that could be done better, where we can probably broadcast in a better way that, okay, we have these, come use it. Yeah.
Okay, thanks, Sharif. I think we need to actually discuss, since we have around 10 minutes left, and this is a topic that could be, we could spend a very long time discussing this, but cross linkages between the different research infrastructures is something that I would like to dedicate the rest of this panel discussion to. So where do you think the priorities are in terms of establishing those linkages? And I'm now talking especially from, let's say, a user perspective. So this is the question. Who would like to comment? Christos, please.
Just a quick comment. There is this experiment that is going on right now in EOSC Future. And I think what we need to do is to go there, see how the research infrastructures collaborate in there, how they make it possible, or they're trying to have, this multidisciplinary, but also cross-domain research, and bring, this experience, back to to BioDT. That's a very important thing, because if we don't do that now at the early stages, we're going, to go ourselves through this exercise, and it's really useless because it's already, done in there. So I think that this is one thing we have, to pay very special attention to.
Okay, so there are existing initiatives where we could go to and see if we can pick up something useful from there. Eric, please.
Cheers. I have worked at the interface between climate science and biodiversity research for a while now. And what I've noticed, and I think this will be relevant for us making these RIs interoperable, is that it all comes down to language and speaking the same language, not just in communicating with one another, but also in how the RIs communicate with one another. And that comes, with the short of it, being wary of the time that we've got left, down to a harmonized metadata standards. I think that's the big development goal here that is also feasible in the time that we have for this project.
Emily, please. Oh wait, you're on mute. So.
Sorry. No, what I wanted to say also was that's true about the standardized metadata. So that's again more technical, but I think from the user perspective, building also a little bit further on what Sharif said, is that for the researchers, I think they just want to know where to get their data and also which services which RI provide. So it's a lot more than just the data points, right? So each RI, like LifeWatch or DiSSCo, also provides a bunch of other services like analysis. So I think from that perspective about cross-linking, I would say is that you need to be able to find your way from one RI to the other if that one would fit your needs better. So I don't know if you could really target towards different disciplines even, and give them like a package of, for your type of research, LifeWatch provides these services, DiSSCo these services. So a really targeted approach, I think, would be interesting. And then also once you are in one of the infrastructures that you easily well, link to the other, if that's where your your research takes you, because in all honesty, I think for the average researcher to know everything about each RI, that would be well, a little bit idealistic. And well, there is a lot of out there, I think, that people are not finding or not aware of at this point. Maybe not GBIF, because that one is already well established, but like LifeWatch or the future, well, DiSSCo also in the future.
Okay, I'm also thinking especially about the BioDT project. Of course, I would say this because I'm in the project myself, but I'm thinking about ways to make these linkages between the RIs work most efficiently from a project perspective. So would anyone like to comment on this? I think very good points, of course, raised already. But
can I just comment on sort of eLTER infrastructures, because we sort of do collaborate with ICOS, so our eLTER sites also are ICOS sites in several cases. So they're cross linkages with that.
okay, existing cross linkages.
Yes, and I mean, for instance, the here in Edinburgh, we have got a site which is linked to eLTER, and it's a global atmospheric watch station, ICOS station, and actors as well. So I mean, these things sort of combined infrastructure to sort of expand the research generally across disciplines is happening at some sites.
Emily?
I just also wanted to say that the link between eLTER and I think DiSSCo will also have to be made at one point, because for example, in my institute, we have soil collections. So I assume these will also go into DiSSCo, and they are well connected to eLTER sites. So I think even there will be cross-linking happening, I hope.
I suppose in the project context, this one point that came up earlier about the deep versus shallow and narrow versus broad is going to be useful for planning. We have just a couple of minutes, so there's still time for a question or comment from the audience, or if anyone would like to ask our panel members, especially about the cross linkages between RIs. I see.
There is a fatigue in the audience.
Yes, okay, we have actually a couple of hands up. Well, Eric being one, but Johannes, do you have a quick question?
No, again, it's not a question, it's more a comment. I think it's the discussion is very is quite interesting. And I think one of the collaborations we are going to do, and BioDT will be really a good example. And Christos already mentioned USC Future. There is also in the EMFRI Fair things ongoing. So these are aspects I think we really can learn. I think BioDT goes one step further, and that is really this piloting, making it happen, also linking to this to the fair provision of data. And I think it's there the interface really would be on the level of data and data services, being it through a service like SOS or a sensor thing, being it an API, whatever. So to enable this programmatic access to information, which was also mentioned by Eric. I think that really would be a very nice use case. I think there are two things important. The one thing is the focus of the research question, and the other thing is the focus of the variables. Because one thing we learned from eLTER, and that's always this issue about this integrative databases overall. It basically it's a very complicated model. You never know what the users want. And once you have set up this integrative database, you'll see that maybe part of it is not fit to the users. So one thing we have to focus on, and that's really will be this interaction, is to have this sustainable data services for a given set of variables. So not take it too broad. Focus really on the things we need. There, I think it's really this linking to the use cases is very important to get this data requirements, the ways how data should be retrieved, and if we do that, we also can agree on technologies, and we really can pilot this. And you know, this would be this interaction points, at least in my understanding, what we could achieve in the BioDT project. And based on that, on top, for example, we can think about on how, and this was also addressed earlier, on how this information, where data are, what the infrastructures are about, so which is more this knowledge web, which is around how this more easily could be brought to the different user communities. So that's also another way forward, and I think this is also what the infrastructures themselves can work on it and really focus on these interactions on the data level for the within BioDT. And
okay, thanks, Johannes. I'm afraid that in interest of time, I need to pass this back to Terhi. So time is slowly running out for the panel discussion, but Terhi, please, would you like to wrap up?
Yes, and do you see me now, or kind of are you back in the general view or not yet?
Not yet, I think.
Okay, this is me also learning to use this. I thought I stopped, but anyway, I could have probably just add myself to the spotlight. Yes, this works. So thank you once more, everybody, for participating in this BioDT research infrastructure workshop. Special thanks for the upright presenters, Sharif and Johannes and Marcella and Christina, and of course also the panel discussion participants. And I think we raised many questions that will be further discussed in the project, and I hope that you have more insights into the research infrastructure that are participating BioDT. And I think this is all that we have for today. We will make the presentation slides available, and hopefully the recordings also for, so if you have colleagues who couldn't participate, so you can let them know. But with this, I wish you a happy continuation for the week, and let's continue the work together. Thank you. Bye-bye.
Thanks, Terry.
Thank you. Bye.
Thanks. Bye.
Very much.
Bye-bye.
Bye, folks.
Thank